Natalie Hastings, Celestial Health. And we are a Cambridge University spin-off company protecting brain networks to stop brain disease with innovative small molecule therapeutics. Our proposition is based on a simple yet impactful hypothesis that in order to treat complex brain conditions such as Parkinson's disease, we need to take a holistic approach and treat the whole brain. We can't cure the whole Parkinson's if half of the brain remains unprotected. And yet, the absolute majority of approved as well as experimental therapies only focus on protecting the type of brain cells called neurons. So what about the other half? Well, in particular, we're focusing on networks of brain cells called astrocytes, which are also fundamental for brain health and become effective in disease. Parkinson's disease currently is incurable. Our target patient profile is our patients with what is known as idiopathic Parkinson's, which means we don't really know what causes their disease. And you might have heard that there has been recent progress with Parkinson's treatments. However, most of them focus on the familial and genetic forms of this condition, which represent the minority of the cases, whilst the majority of the patients are still underserved. The patient is usually diagnosed as they present with a range of motor as well as non-motor symptoms, and they end up with a range of symptom masking drugs, which often cause serious side effects, such as hallucinations and involuntary movements, which lead to more symptoms. And in the meantime, the disease keeps progressing and the patient keeps getting worse. Parkinson's is a neurological condition with the highest rise in prevalence worldwide, which is largely linked to the aging population, as the number of people over 60 is predicted to almost double from 2015 to 2050, and that is exactly the population where Parkinson's is most prevalent. There are up to 10 million people with Parkinson's worldwide, and over 150,000 in the UK alone. In the US, the economic burden of Parkinson's is predicted to exceed almost 80 billion by 2037 due to direct treatment costs as well as carer costs and loss of income. The global Parkinson's market is predicted to exceed $8 billion by 2026. And we're expecting to launch our new drug around 2040, when the estimated market in Parkinson's for small molecule therapeutics is predicted to be around $12 billion. So our strategy is to slow down or stop the progression of Parkinson's disease at the time of diagnosis with our innovative small molecule drugs, which are safe and brain penetrant. So as we discussed, the number of etiologies or factors converge to cause idiopathic Parkinson's, which is exemplified by the astrocytic network disruption. And therefore, we propose that with our drugs, we can stabilize the astrocytic network and prevent the development of symptoms in a range of function-specific neuronal networks. The first target of our platform is a protein called connexin 43. It sits at a unique nexus where it's known to be selective to astrocytes in the CNS. It is also known to be druggable and safe, as there is a number of companies globally who are working on this target, mainly in the periphery, because of the poor brain permeability of their assets. But nevertheless, this suggests that we can safely alter the biology of this protein. And this target has been implicated in Parkinson's, and particularly by us, which was the focus of my background academic work at the University of Cambridge. So why connexin 43 disruption matters in Parkinson's? Under conditions of cell stress, such as those found in Parkinson's, which include inflammation and aggregation of alpha-synuclein, astrocytes uncouple and they no longer form a network, which then leads to inflammatory dysregulation in the brain which then further promotes abnormal signaling and aggregation of alpha-synuclein. With our treatment, we can stabilize the astrocytes so the signaling between the cells can flow in a more healthy manner, which then reduces the abnormal inflammatory environment and also prevents abnormal alpha-synuclein spread. So therefore, we're proposing, based on our preliminary data in cell and animal models, that the astrocytic network dysregulation is an upstream target compared to other experimental disease-modifying approaches. As with our new drugs, we can prevent the inflammasome activation, abnormal protein aggregation, abnormal mitochondrial function, and ultimately cell death, which then lead to symptoms. This approach is unique in the field, as there has been increasing interest in the field of Parkinson's and inflammasome inhibition, so the anti-inflammatory and immunomodulatory approaches. The advantage of our system is that our drugs do not come with the side effects of potential immunosuppression, which could have side effects, especially under the chronic conditions in the elderly population. Also, astrocytes are gaining prominence in the field of Parkinson's. However, the approaches to drugging astrocytes remain purely academic. So as I mentioned, we are a spin-off from the University of Cambridge, where we conducted initial studies to validate the target and prove the concept with tool compounds. Celestial is now fully spun out and secured an exclusive IP license from the University of Cambridge, and we have now closed our pre-seed round, which was slightly oversubscribed. The goal of the pre-seed round is to create and initially characterize the new drug leads, of course, in collaboration and support of O2H, as well as another key partner, Charles River. And soon we will be opening our seed round, which will allow us to complete the preclinical development of the chosen leads. And we're actively seeking feedback from potentially interested funders. The team is led by myself. I have over 10 years of academic experience in clinical research and bioengineering. We have engaged a chairman, Stephen Grant, who is based in Boston, USA, to engage with the Boston ecosystem, where I will be next week. We're also joined by the investor director from Zinc, Natalie, and my technical colleagues, Saifa and Alex, are helping with the pharmacology and chemoinformatics side. We're advised by a number of scientific leaders in Parkinson's, preclinical and clinical development, and also the business and commercial expertise is provided by our business advisory board. At the moment, our key series of assets, in fact, it's actually several chemical series of assets in development and discovery, are focused on Parkinson's, but we're also looking to expand our indication to our pipeline to new indications as well as to new targets, because astrocytes are fundamental in brain health and become disrupted in a whole range of diseases. Therefore, there is a great scope to make a difference to patient populations in other conditions, including, but not limited to, multiple sclerosis, depression, and other dementias. At Celestial, we're bringing together the expertise in astrocyte biology, efficacy, screening assays, and therefore we are looking for your support, we're looking for your feedback, and if you would like to hear more, discuss any of these aspects, we would love to hear from you.
Thank you. So one of the biggest challenges in Parkinson's disease is to really identify the right patient populations and the time of intervention. Given that we replace now the hypothesis of the astrocytes early upstream, how are you thinking about identifying those patients that would respond to that therapy early on, given that there is no biomarker available now to identify them?
I think there are two parts to this question. Thank you very much for this. Firstly, we are working on biomarker identification as part of our activities, and we have a collaboration with the University of Manchester for that purpose. We're working on CSF, which is accessible in clinical trials. But also, secondly, I mentioned that this is a unique target in which a number of conditions or etiologies converge, which means we're actually not limited to a very specific patient population, which is defined by a specific mutation. If they don't have this mutation, the treatment is not going to work. In fact, I've seen that myself in cultures. It doesn't matter what the lesion is, whether it's interleukin, alpha-synuclein, or even mechanical injury or hypoxia. All of those factors converge in the disruption of this target. So that means we have a broader scope for the patient populations who are likely to respond to this treatment.
So your platform is basically Cellestial.
So the platform is more of a conceptual platform, and it combines a few approaches. So there is the bioinformatics approach to establish the druggability and astrocyte selectivity. And the assays, the screening assays, so it was developed or is being developed and optimized specifically for this target. For another target, there might be a new screening assay.
Thank you so much.